In this video, we will discuss about a paper that talks about the pancreatic cancer stiffness, whether it varies with the cancer cell progressions or not. So this is what they wanted to check. So the first figure talks about in the first figure they have they have used three different models at different stages of pancreatic cancer. First one is the healthy pancreas, and second one is the PanIN, which is the early stage of the pancreatic cancer, and third one is the KPC mice model, which is the aggressive stage of the pancreatic cancer or pancreatic ductal adenocarcinoma. So first, and they did their studies in the mice model. So from that, once the cancer is developed, they stain the cells for this Sirus red. From that, they will get an idea about the collagen fibers that are formed across these different models, and they are able to see the collagen formations seems to be high and are more aligned in the PDAC case than the PanIN or normal case. And along with that, they also check the thickness of this collagen fiber, and what they are able to see that in the normal pancreas the thickness is lower, and when they move to this PanIN condition, the thickness increases significantly. However, when from the PanIN to PDAC conditions, the thickness does not increase that much. So from that day, it seems like this collagen fiber formations or the fiber formations are usually occurs during the early stage of the pancreas. Thereafter, they also checked the alignment of this collagen fibers, and the alignment of the collagen fibers shows that the alignments is highest as the pancreatic cancer progresses, and thereafter it goes. Thereafter, the PanIN comes into place, which has the intermediate alignment, and normal pancreas has least alignment. And this can be interpreted in a way that as the pancreatic cancer is become more aggressive in nature, then it will get in aligned. In order to be more metastatic, it needs to be aligned with the surrounding tumor microenvironment or this collagen fibers in this case. So as the cancer progresses or becomes more aggressive, it becomes more aligned with the collagen fiber. This could be interpreted from this result so far. Thereafter, they check the pancreatic cancer tissues, this young modulus measurement using the AFM. The schematic is shown in the panel B at how they measure this young modulus using this atomic force microscopy. So they have used this 70 micromolar ball and then roll around it to check the pancreatic tissue young modulus across the region. And what they are able to see that there is a variation in this stiffness. So there is a variation in this stiffness in all the three models. However, this variation in stiffness is narrows down as they move from normal pancreas to PAN, as they move from PAN to PDAC. That can be seen from this figure 3. Thereafter, when they check the mean values of this young modulus of the pancreatic tissues in three different models, they are not able to see significant difference. However, when they check the median values of this young modulus, they are able to see significant difference that can be seen in this panel D1. However, for this mechanotransductions, the stiffer tissues usually drives those mechanotransductions forces. So thereafter, they also checked they also compared the upper quartile of this young modulus of this pancreatic tissues, and from that, this also shows a significant difference. That is, PDAC shows a values of around 4 kilopascal, whereas PanIN shows the values of around 2 kilopascal, and the normal shows the values of around 1 kilopascal. So this also indicates towards the point that as the pancreatic cancer progresses, it becomes the stiffness across these pancreatic tissues, across these pancreatic tumors become more stiffer, and that also helps in the mechanotransductions, and this could somehow play a role towards this pancreatic cancer stiffness. or the pancreatic cancer aggressiveness. So this is what so far it looks like from the results represented in this figure 1. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.